Hey, NAMI students, especially us again. We're going to do day 2 of chapter 17, general or sense organs. Okay. So I want to wrap up the general ones first. So I probably should have done this slide in the first video, but whatever, I didn't get done. So receptor function. Now, what's fascinating is you got to convert these stimuluses like sounds, smells, whatever, into a nerve impulse, send it to your brain, and your brain has to go through all the files and figure out, what am I looking at? What am I hearing? What am I smelling? Right? So there is memory tied in with that. It is an all or nothing response. Once you get a stimulus triggers this, it's going to go. Now it does adapt slowly. Um, some adapt rapidly, some adapt slowly. Like, you got receptors in your deep in your body, like you sprain an ankle, you pull a muscle. They're going to send stimuluses to you to tell you, hey, you got an injury, stay off of that, be smart, right? Other ones, like your fingers and touch receptors, they adapt very quickly. So you may still be holding on to something, but eventually you may not know it's there because it just quits responding. I had a funny one the other day. I'm looking all over for my sunglasses, and my wife starts laughing at me and says, they're on the top of your head, right? Because they were up there and they just quit responding, and I forgot they were there. So that's what we're talking about. Pretty simple, but that's general ones. Okay. Now we're going to change gears a little bit. Let's talk about a special sense organ. We're going to dissect the eye in lab. So the eyeball is a very fascinating structure. It's got lots of parts, and we use the eyes for a lot of new learning. We talked about that in the brain chapter. 95% of what new learning comes through our eyes, right? So the sclera is the outside part of the eye, the white of the eye. There's no blood vessels found in parts of that. The cornea is a clear covering over the front. And then we'll talk about the canal slim down the road in notes. But if you look at a diagram, you can see this is all the sclera, kind of three-fourths the way around. And right in front, the sclera is transparent, and that's called your cornea. All right. Um, Not a lot of blood vessels in part of that, so you can get poked in the eye and doesn't necessarily bleed. Now, there is some blood vessels inside the eye, so if you go very deep, you're going to get into blood, but the outside doesn't have a lot. Okay, has feeling, if you get poked in the eye, but not really so much blood vessels. Okay, the choroid layer does have a lot of blood vessels. It's got three parts. You got the ciliary body. You can see that in red right here. We got the ciliary process in green. And then we got these little suspensory ligaments that hold the lens in place. So we got lots of parts to that. So that's called the middle layer of the eye. This will make more sense when we dissect it, right? The ciliary muscle is actually smooth. It's actually a muscle. It's smooth, and it does help change the shape of your lens. Sometimes eye size, you can tell with mine. I have to use reading glasses because you lose that elasticity and your lens doesn't change shape like it used to when you were younger, and you have a hard time seeing stuff up close. All right. The suspensory ligament that holds your lens, keeps it from floating. The iris, that would be the colored part of your eye, and it has a donut shaped mass in the middle called the pupil. Okay, you can see that. And again, you'll see that when you dissect as well. So there actually is a hole in here. You can see it right here where the red arrow's at. So the iris is coming down from the top and bottom, and there's a hole in the middle. Um, and that is your pupil that will change shape as bright light and dim light comes in so that you can see in those situations. And then the very back of the eye is called the retina. Three layers of that. We'll get into that a lot more when we talk about the physiology, how we see. And the main parts of that is called cones and rods. So you'll see that a lot as we dissect and get through it as well. Okay. Cones are less numerous than rods, and they're most densely concentrated in the fovea centralis, which is this area of there's your cones and rods. It's not what I wanted. There you go. This is the fova centralis. It's that little tiny spot. That's your sharpest vision. Isn't that crazy? And then this big section over here, which again you'll see when you dissect, that is the optic nerve. That's your blind spot. Okay. So that's really kind of the main parts of the eye. We'll kind of break that down and go over that a little bit more as we go through how the eye functions. We'll talk about the cavities and the humors. Your eye is not solid, it's filled with fluid and gel. And we'll talk about that stuff in the next video. All right. Hey, have a good day. Pretty short one today, but I just want to get the main parts of the eye in, and you'll see more of that when we dissect it. All right. Hey, have a good day. See ya.